Hello everyone, welcome you all in this video lecture. So in this video tutorial, we are going to discuss about an important class of bacteria which are called as gram-positive bacteria. So let's start with the introductive part. So before moving to the main topic and learn about the gram-positive bacteria, let's understand about some characteristic features about the bacteria. So the bacteria are one of the oldest life forms in the earth, which means that bacteria are considered as one of the oldest living organisms in this earth. They are prokaryotic and unicellular organisms. They are single-celled organisms which are microscopic and can be seen only with the help of electron microscope. So the bacteria have prokaryotic composition because they don't have a well-defined nucleus and also lack some important cytoplasmic organelles which are called as membrane-bound organelles, that are mitochondria, endoplasmic reticulum and many other organelles which are found in the eukaryotic organisms or in eukaryotic cells. So that's why they are considered as prokaryotic organisms. Now these bacteria are categorized into different types on the basis of shape, cell wall composition and oxygen requirement. So for example, according to the oxygen requirement of particular bacteria, they are divided into two types which are called as aerobic bacteria and anaerobic bacteria. So similarly, on the basis of the cell wall composition, the bacteria are divided into two types which are called as gram-positive bacteria and the another type is gram-negative bacteria. So in this video lecture, we are going to discuss about the gram-positive bacteria, their structure, their cell wall composition and their functions in detail. So let's start with the topic. So let's understand about some characteristic features about the gram-positive bacteria and why they are called as gram-positive bacteria. So the gram-positive bacteria are the genus of a bacterial family and they are a member of phylum Firmicutes. So basically, according to the bacterial nomenclature, they are divided into different phylum and the gram-positive bacteria are belong to the phylum Firmicutes, and they are those type of bacteria which retain crystal violet dye in the differential gram staining. So basically the differentiation between gram positive and gram negative is defined with the help of a particular staining method which is called as gram staining. And there are several examples of gram positive bacteria which are like Actinomyces, Clostridium, Mycobacterium, Nocardia. All these bacteria are pathogenic and cause different diseases in the humans and other higher animals, and they belongs to gram-positive class of bacteria. So let's understand some basic facts about gram staining and what is the gram staining. So basically it is the most crucial staining method used in the field of microbiology to make the differentiation between bacterial families or bacterial genus. So this method was first discovered by Hans Christian Gram in 1882 and the method was also named over it as gram staining or gram positive and gram negative bacteria. So the method involves the use of crystal violet and methylene blue to stain the particular bacterial organisms, and after the staining, according to the results, they are named as gram positive and gram negative bacteria. So the organisms which retain the primary color and appear purple brown under the electron microscope are considered as gram positive bacteria, and they are differentiated according to their cell wall composition because both the bacteria have different cell wall composition. That's why some of them retain the primary color and appear purple brown and another some can't retain or can't resist the primary color and they appear pink or red colored organisms. So the organisms which do not take the primary staining and appear red under the electron microscope are called as gram-negative bacteria. So now let's discuss about the characteristic features of gram-positive bacteria. So as we have discussed in our earlier slides that the differentiation is done according to the cell wall composition of both the bacteria. So in the case of gram-positive bacteria, the cell wall is composed of thick peptidoglycan layer and a cytoplasmic lipid membrane. And these bacteria have a thick peptidoglycan layer as compared to the gram-negative bacteria, and also they have higher content of teichoic acid in their cell wall. Now the gram-positive bacteria have low lipid content in the cell wall, but in the case of gram-negative bacteria, they have a higher content of lipid in their cell wall. So the peptidoglycan layer of these bacteria play a major role in maintaining the rigidity of the cell wall and by cross-linking the assistance of transpeptidase. So now let's understand about the cell wall composition of gram-positive bacteria. So the cell wall of the gram-positive bacteria includes three main components in its cell wall. The first one is peptidoglycan, which made the peptidoglycan layer, and the second component is lipid, and the third one is teichoic acid. So the peptidoglycan is also called as murin, and that's why it is also considered that gram-positive bacteria is made up of murin. So this peptidoglycan or murin made 90% of the bacterial cell wall content, and that's why they are differentiated by gram-negative bacteria. So its major role is to provide rigidity and shape to the cell wall. Now it have the scientific name, and it is made up of two sugar derivatives which are named as N-acetylglucosamine, and the second one is N-acetylmuramic acid. These two components are made peptidoglycan layer of the cell wall. The second component of the cell wall is lipid layer. So there have a thin layer of lipid. In the case of gram-positive bacteria, it is only about 2 to 5% of the total content of this cell wall. And the third component is teichoic acid. So it is made up of a polymers of glycerol, and it is totally soluble in water and make 50% of the bacterial cell wall. So these are three components of the cell wall, and these three components made the gram-positive bacteria's cell wall. So now let's understand the cell wall composition of gram-positive bacteria with the help of this diagram. So here in this diagrammatic presentation, we can see this is the structure of cell wall of gram-positive bacteria. So we can see in the uppermost there is a layer of teichoic acid and lipoteichoic acids. So there is a thick layer of peptidoglycan which have teichoic acids in its structure also. And this peptidoglycan layer is followed by a periplasmic space. So basically the periplasmic space is the space between the cell wall and plasma membrane of the cell. So this is called as a periplasmic space, and this periplasmic space is followed by the plasma membrane or cell membrane of the cell, which is a bilayer membrane of several proteins, lipids and carbohydrate molecules. So this is the structure of gram-positive cell wall, and these are the main components, which are lipoteichoic acid, teichoic acid, peptidoglycan and a periplasmic space in between plasma membrane and cell wall of the cell. So now let's discuss about the shape of gram-positive bacteria. So basically the bacteria can be found in different shapes, and according to their shapes they are also divided into different types. So the gram-positive bacteria can be found into cocci or bacilli, two main forms. So the cocci or the spherical forms, which are can be single or can found in the chains, and the another form is bacilli, which are rod-shaped bacteria. And there are two other forms of gram-positive bacteria, which are tetrad, which is a type of cocci shape which occur in square clusters of 4. The example of tetrad is Micrococcus species, and the fourth form is Sarcina, which have a thick-walled cocci shapes, or it is also shaped as an octade. So these are the main shapes in which gram-positive bacteria can be found. So now let's discuss about the effects of gram-positive bacteria. So basically these bacteria can affect in both the types. They can also affect with some benefits and also cause several diseases in humans. So let's discuss about the benefits of gram-positive bacteria for humans. So basically there are several species of gram-positive bacteria which are non-pathogenic or which act as a probiotics. So the probiotics are the class of bacteria which are beneficial for humans, and these species resides in our mouth, skin, intestine and upper respiratory tract, and they are not pathogenic for us. Several species of gram-positive bacteria are also useful in the production of Swiss cheese and also in many other bakery products. And a species named as Bacillus amyloliquefaciens is a very good source of antibiotic protein Bernaceae. So it is used in the medical field for the antibiotic production. So these are some effects or benefits of the gram-positive bacteria for humans. So beside the benefits of gram-positive bacteria, several species of gram-positive bacteria are also harmful, and these species cause several harmful diseases or infections in humans as well as other higher organisms. So there are some examples or some disease caused by gram-positive bacteria like Streptococcus pyogenes, which is also a species related to gram-positive bacteria, and it causes rheumatoid fever in humans. And another species of the same genus, Streptococcus pneumonia, causes bacterial meningitis and bacterial pneumonia in human, which is a lung-related disease. The third species, named as Clostridium botulinum, causes botulism or poisoning, which is associated with visual disturbances, also with respiratory paralysis and many other serious problems. The same species, Clostridium tetani, is harmful also and also caused tetanus in humans, and a disease caused as by Listeria monocytogenes, which causes bacterial meningitis in humans. So besides these, there are several other diseases like typhoid or many other diseases which are caused by gram-positive bacteria. So in this video lecture, we have discussed about the gram-positive bacteria, their cell wall composition and effects which can be harmful or beneficiary effects of gram-positive bacteria for humans as well as for other higher animals.